So friends, next class of the phytohormone or plant growth regulator or plant hormone is brassinosteroids. Now what we are going to learn about brassinosteroid is that what are these brassinosteroids, how they came into existence, how they are biosynthesized in the plant, what is the basic pathway that is followed in their biosynthesis, how they exhibit the effect on the plant system, that is their signaling mechanism, what are the different components what are the different proteins that are involved in that signaling mechanism. And in the end we will discuss about the physiological effects that are exhibited by these brassinosteroids on the plants. So first, what are brassinosteroids? So as their name indicates, steroids, these are the steroidal molecules having a cyclopentanophenanthrene nucleus, and some of the steroids have modification in this cyclopentanophenanthrene ring. Obviously a side chain is also attached to this nucleus, and since these are the derivatives of the basic cholesterol nucleus, these are also known as 5-alpha cholestyne derivatives, and depending upon the number of the carbon atoms that are present in the structure, they can be categorized into C27, C28 or C29 brassinosteroids. So if there are 27 carbons or 28 carbons or 29, they can be categorized on these basis, and this variation in the number of the carbon is largely due to the variation in the side chain that is attached to the steroidal nucleus. And as I told you that all the brassinosteroids have a steroidal nucleus, and some have an heterocyclic ring B with a side chain on a 17th carbon. So as you can see, there is a heterocyclic ring B, a ketonic group is also attached, oxygen is also present in the ring B. There are certain common features that have been seen in almost all the brassinosteroids. The carbon 18 and carbon 19 methyl groups have a beta angular orientation on the structure. The hydrogen atom which is present at the junction of ring A and B at 5th carbon has an alpha orientation. So these are the few features which are commonly observed in almost all the brassinosteroids. Then the hydroxyl groups that are present at 2nd carbon and the 3rd carbon in ring A and carbon 22nd and 23rd of the side chain, all these hydroxyl groups are present in the alpha orientation. Now we'll move on to how these brassinosteroids were discovered. It was Mitchell along with his associates in 1970 at United States Department of Agriculture and Plant Hormone Laboratory, Maryland. They reported that extract of pollens that were obtained from the rape seeds, that is Brassica napus, and elder, that is Elnus glutinosa, they have an unusual effect on the growth of the plant. So eventually, though the compounds that were responsible for such kind of effect were not elucidated, were not identified, were not isolated, they believe that certain lipophilic compounds are present in those extract, and they gave a name brassin, because initially that extract was developed from the pollens of Brassica napus. So they gave the name brassin to that extract that was responsible to produce the unusual growth response. Later in 1972, Mitchell and Gregory, they showed that brassins could enhance the crop yield. Not only the crop yield, but they also exhibited that it enhances the efficiency of the crop against various stress conditions. Brassins also improve the vigor of the seed. So these were the few effects that were exhibited by brassin. Then in 1979, Grove and his group, they were the person who first isolated the compound from that extract of pollens of Brassica napus, and they named that compound as brassinolide. So we can say that brassinolide was the first brassinosteroid compound that was isolated from the plant. And since first the compound was isolated, brassinolide, it opened the door for future discoveries, and the scientists start again working on such kind of compounds. And in 1982, castasterone, another type of brassinosteroid, was isolated from the insect gall of chestnut. So this was the second compound that was isolated, and this is how basically the brassinosteroids came into picture and their effect on the plant growth. Now these brassinosteroids are found in gymnosperms, they are also found in mono and dicotyledonous plants, they are also found in algae, and the highest concentration of brassinosteroids is normally found in the reproductive organs and the tissues that are growing, and these reproductive organs can be pollens, growing tissues can be mature seeds or the shoots. Now we move on to the biosynthesis of brassinosteroids. Now campesterol has been considered as the precursor of brassinolides or brassinosteroids. Though the campesterol is considered as precursor, but it all starts with the mevalonic acid pathway. Mevalonate gets through a series of steps, it gets converted into squalene, squalene gets converted into cycloartenol, cycloartenol through numerous steps gets converted into campesterol, and campesterol then it is reduced to another compound known as campestenol, which undergo hydroxylation to produce 6-hydroxycampestenol. This 6-hydroxycampestenol undergo oxidation and produces 6-oxocampestenol, which further undergo hydroxylation to produce cathasterone. So here you can see mevalonate is there, through series of steps it gets converted into squalene, squalene through series of steps gets converted into cycloartenol, cycloartenol through various steps gets converted into campestenol, campestenol eventually gets converted into 6-oxocampestenol, which converts into cathasterone. Now cathasterone through various intermediates, specifically T-sterone, tiphasterol and castasterone, gets converted into brassinolide, and this is how brassinolide is produced in the plant matrix. Now the question is how these brassinosteroids produce effect on the plant system. They have a signaling pathway, and before we discuss about the signaling pathway, we will first discuss about various components that are involved in the signaling pathway of brassinosteroids, and these components are nothing but different types of proteins that are present in the plant system, in the cytoplasm or in the nucleus or on the membrane, that exhibit or that initiate the signaling pathway of the brassinosteroids. So the first component is the leucine rich repeat LLR membrane bound receptor, which is serine threonine kinase type, and it is also known as brassinosteroid insensitive 1, that is BRI1. So in simple words we can say BRI1 are nothing but the receptors that are present on the membrane which causes the binding of brassinosteroid onto the plant cell. Second component is the BRI1 kinase inhibitor or BKI1. Third is the another receptor kinase, BRI1 associated receptor kinase or BAC1. This is another, I will say, a protein or a receptor molecule that is present on the membrane of the plant, and as soon as the brassinosteroid molecule attaches with the BRI1 receptor, the BAC1 gets stimulated and they become active, and they further take away the signaling pathway, as we will discuss further in the diagram. Next component is the BR signaling kinase or BSK1. Protein phosphatase molecules are there, and their related protein BRI1 suppressor, that is BSU1 protein, which is responsible for the exhibition of the brassinosteroid responses, and eventually certain protein, it activates certain protein which enter into the nucleus of the plant cell, causing initiation of the gene expression. Then we have brassinazole resistant 1, that is BZR1, and brassinosteroid insensitive 1 EMS suppressor or BES1 or BZR2 proteins. These are the components which enter into the nucleus, which are get activated by BSU1 protein, and these two components enter into the nucleus, and in the nucleus they further activate brassinosteroid response element, that is BRRE elements, which upregulate the expression of the genes that exhibit or that are responsible for exhibiting the brassinosteroid mediated effects on the plant. Now how it happens? Now when brassinosteroids they bind to the extracellular domain of the BR insensitive 1, or we can say brassinosteroid receptors. So once these brassinosteroid molecules bind to these receptors, they promote or they stabilize the formation of BR1 homodimers. So dimer of these receptors are formed, and as I told you that BRI1 kinase, it also gets activated. Now BRI1 phosphorylates the inhibitory BRI1 kinase bound protein, or BRI1 kinase inhibitor, or BKI1, and induces its dissociation. So it gets dissociated from the receptor complex. Now once this BKI1 dissociates from the receptor complex, it causes the association of BRI1 with the second receptor kinase, that is the BAC1 receptor kinase, which is also present on the membrane of the cells, as we have already discussed. Now this binding of BAC1 to the BRI receptor complex causes series of autophosphorylation and transphosphorylation between BRI1 and BAC1, which further activate the BRI1 kinase that triggers the downstream BRI signaling kinase, or BSK1, through phosphorylation. And once the BSK1 gets phosphorylated, it dissociates from that complex, receptor complex, and binds to protein phosphatases. These protein phosphatases further activate BRI1 suppressor 1 proteins, or BSU1 proteins. Now these BSU1 proteins are important because they cause dephosphorylation of certain GSK3 or Shaggy or F-box kinases, which are known as BIN2 proteins. Because these BIN2 proteins, as we will discuss ahead, they are responsible for the degradation of the nucleic factors that are responsible for the BR stimulated actions. Now this BIN2 proteins, in the absence of brassinosteroids, as I told you, they are responsible for the phosphorylation of BZR1 and BES or BZR2 proteins, which are the nuclear factors and are responsible for the expression of the genes. So once these nuclear factors are phosphorylated by BIN2 proteins, they become more susceptible towards the proteasomal degradation through protein 26S proteasome proteins. But when brassinosteroids are present, BSU1, by the action of protein phosphatase, it inhibits the BIN2 proteins, and this inhibition causes the movement of BZR1 and BZR2 proteins into the nucleus, thereby causing the exhibition of the brassinosteroid mediated actions. So once they enter into the nucleus, they bind to brassinosteroid response elements to cause the expression of the genes that are responsible for brassinosteroids action. So here you can see in this picture, it is divided into two parts through a dotted line. The upper part is the condition when brassinosteroids are absent. So you can see that the receptor complex, to the receptor complex BSK1 is attached, then another second receptor kinase is there, that is known as BAC1, and to the BRI1 kinase or the receptor BKN1 is attached. So as soon as, since the brassinosteroids are absent, you can see that BIN2 is phosphorylated, and this phosphorylation of BIN2 proteins further causes phosphorylation of BZR1 and 2, and once these are phosphorylated, they become more prone towards the proteasomal degradation, and they get degraded by the 26X proteasome proteins, and no effect is exhibited. But when brassinosteroids are present, that is the second scenario, you can see that brassinosteroid molecule binds to the extra membranal or extracellular part of the BRI1 receptors. And once these brassinosteroids are bound to the receptors, they bring certain conformational changes in the receptor, causing the binding of BAC1, which is the second receptor kinase. So a complex is formed, and the third component is the BSK1. So this receptor complex, which is composed of three parts, BSK1 and BAC1 and BRI1 receptors or the proteins, they undergo phosphorylation, and with the help of this phosphorylation, BIK1 protein gets dissociated from the receptor complex, because this is an inhibitory protein or the negative regulator of brassinosteroids. And once this inhibitory protein is separated from the receptor, the downstream signaling pathways or mechanisms, they become more active. So this BSK1, it also undergo phosphorylation, and once it is phosphorylated, it reacts with the protein phosphatase or PP2A molecule. This phosphorylation and reaction with PP2A causes stimulation of BSU1 proteins. And I told you that BSU1 proteins are important because these are the proteins which inhibit the action of BIN2. As you can see, BIN2 are responsible for the phosphorylation of BZR1 and 2, which are the nuclear factors which enter into the nucleus to exhibit brassinosteroid mediated action. So once BIN2 proteins are inhibited, BZR1 and R2 are free to enter into the nucleus. And once they enter into the nucleus, they exhibit a positive effect on brassinosteroid response elements, and these response elements further upregulate the expressions of the genes that are responsible for the brassinosteroid mediated action on the plants. So this is how basically brassinosteroids exhibit effect on the plant. Now question is, what are the different effects that are exhibited by brassinosteroids? First one is, brassinosteroid causes elongation of the cells. It has been observed that application of extracellular brassinosteroids causes elongation of hypocotyls, apocotyls, peduncles of dicots, as well as cleopetyls and mesocotyls of monocots. And this effect of elongation, cell elongation, is largely attributed to the cell wall loosening effect of the brassinosteroids. Though the action is similar to auxins, but the action differs in the term of the kinetics. Auxins has a very short onset of action, or we can say very short lag time, but the effect, maximum effect, lasts up to 30 to 45 minutes. Whereas in case of brassinosteroids, the onset is slow, or you can say the lag time is somewhat around 45 minutes, but the effect remains for few hours. So short onset of action in case of auxins, and the effect is short lived. Whereas in brassinosteroid, onset of action is slow, but effect remains for longer duration. Then brassinosteroids also stimulate cell division, and not only they stimulate the cell division, but also the rate of the formation of the new cells. They also cause the differentiation of the protoplast and formation of the cell wall during the cell division, which eventually increases the number of the cells or the viable cells, because once these brassinosteroids help in the formation of cell walls, the viability of the cells which are formed increases, and this is how the rate as well as the cell division increases. Now brassinosteroids have also been observed to cause vascular differentiation, especially the tertiary elements or the xylem vessels. It has been found that they upregulate xyloglucone endotransglycolase enzyme, or the genes that are responsible for the action of these enzymes, and it has been found that this enzyme is responsible for the formation of the cell wall, strengthening of the cell wall vis-a-vis. In other words, we can say that it helps in the modification of the cell wall from the soft one towards the harder one. So this is how they causes the differentiation of the vascular tissues as well. Then, as I told you, that the highest concentration of brassinosteroids is found in the reproductive organs and pollens, because the discovery of the brassinosteroids were for pollens also. It has been found that brassinosteroid causes the elongation of pollen filament, thereby causing the inhibition of the male sterility or the regulation of the male sterility. In many cases, it has been found that the length of the pollen filament, if it remains short, it's very difficult for the pollens to reach to the stigma, and in numerous cases it has been found as a major cause of the male sterility in the plants, or the male sterility or the sterility of the male flowers, because the pollen filament is quite short and pollens are not able to reach to the stigma. So by the application of brassinosteroids, it has been seen that the length of the pollen filament increases, thereby controlling the male sterility or improving the male fertility. And it's not only that the length of the pollen filament is increased, it has also been observed that application of brassinosteroids increases the number of the pollen grains that are produced, vis-a-vis it also increases the quality of the pollen grains that are produced by the flowers. Brassinosteroids also help in the development of haploid seeds, thereby producing genetically more stable plants. These steroids have also been found to stimulate the production or the synthesis of nucleic acid and proteins, which eventually help in the overall growth of the plant. Brassinosteroids have also been found to increase the resistance of the plant towards various kind of stress condition, whether it can be a biotic or abiotic stress or pathogens which can be of bacterial, fungal or the viral origin. Now the brassinosteroids have shown to stabilize the plant membranes, thereby preventing the leakage of different electrolytes, vis-a-vis also increase the synthesis of ATP, and most important is increase the production of antioxidant components in the plant cells. So stabilizing the membrane, prevention of the leakage of electrolytes, increased ATP and antioxidant production, increase in antioxidant production, all these factors help in making the plant cells able to fight towards various kind of biotic and abiotic stresses or pathogen. So this is how I will say brassinosteroids, they exhibit the protective effect on the plants, or they make the plant able to protect themselves against different kinds of pathogens, whether it's a bacterial, fungal or viral, vis-a-vis also the stress, abiotic or biotic stress. Now brassinosteroids have also shown to increase the yield of the crop, and not only the yield but quality of the crop as well, and this quality can be determined in the term of the size of the grain, weight of the grain and the number of the grains that are produced by the plant. Quality can also be assessed in terms of improved nutrient composition of those grains or the seeds that are produced by the plants. So friends, this was all about brassinosteroids, that how they were discovered, how they are biosynthesized in the plant, what are the various signaling pathway, how they exhibit the effect on the plant through various protein molecules that are present either on the membrane or in the cytoplasm or in the nucleus, and how these proteins get activated or deactivated in the presence or absence of brassinosteroid, how these presence of brassinosteroid lead to the activation of certain nuclear factors which causes activation of brassinosteroid response elements, and these response elements how they further activate the genes or upregulate the expression of the genes that are responsible to mediate the effect. And in the end we discuss what are the different physiological effects that are exhibited by the brassinosteroids. Thank you.